Thank you very much. So I'll present our current work in progress on whole brain dynamical modeling of adolescent developing brain. So this is not published, so I look forward to your feedback. Okay, so I'll cut to the chase why we did this study. We were interested in studying how excitation and inhibition matures during adolescence in humans in vivo. And why do we care about this? It's because, so we have these two forces in the cortex, excitation and inhibition, and they need to work hand in hand and be in sort of a state of balance so that they enable many of the normal cortical functions. And it's also important to note that both excitation and inhibition undergo a lot of changes during lifespan, and importantly during adolescence. And the maturation of excitation and inhibition during adolescence is very important for normal development. So much so that it's suggested that their impaired development during adolescence can lead to psychopathology, importantly schizophrenia. Okay, so the problem is that much of what we know about development of excitation and inhibition comes from animal studies. It's because we can put electrodes in the brains of animals and then measure the activity of their neurons, right? It's easier to study them in animals, but it's not much easier to study them in humans as well, because we cannot do this in humans. Instead, what we have available in humans is usually more large-scale methods, such as fMRI, but they don't have good enough spatial and temporal resolution to enable studying this excitation and inhibition at the micro scale. So the question is how we can relate what we have, brain imaging, to what we want to know about excitation and inhibition. And here, computation modeling comes as a bridge, which is usually used when you want to measure something that is hidden, but then on the other hand, we cannot measure that. On the other hand, we have something that we can observe. And the good news is that we also know some of the rules that relate what we cannot measure and what we can measure. And we can model this. And then we can modify our model until it produces something that is similar to our observation. And then we can use this model to study the hidden feature that we're interested in. So that's exactly what we did here. We used this approach of whole brain dynamical modeling, or neural mass modeling, on a data set of imaging data from adolescents to study how excitation inhibition and cortical microcircuits develop during adolescence in humans. Okay, so here's how we did this. We started with a data set, with an imaging data set of adolescents, Philadelphia neurodevelopmental cohort. We included 627 subjects aged from 10 to 19, then processed their images using FreeSurfer, fMRI Prep, and Mika pipe. And then, using their images, we built individualized models of their cortices to get assessments of their EI ratio status, and then looked at age variation of these estimated EI ratios based on the model. Okay, so I'm going to now go into the more detail of how we built these individualized models. We start with a very simplified model of the cortex, where we have 100 nodes, Schaeffer 100 prosylation if you know it, and then connect them together based on the actual structural connectome of the very subject that we want to model, right? And then zooming in on each of these nodes, in our model we have two populations of neurons, or two neuronal ensembles, excitatory and inhibitory. And these are connected to each other based on a certain, based on given local weights. But also, you can see that the excitatory units of different nodes are also connected to each other through the structural connectome of this subject. As you can see here, I have highlighted with red some values, some weights, and these weights are the actual parameters of our model, which we can control, and through controlling these parameters, we can personalize the model towards to fit our subject's data. We had two cases here, or two alternatives for modeling. One case was a homogeneous model, in which we assume that the local weights, starting with the W, are the same in every node. This is easier to fit because it has a small number of parameters. But also is not very realistic because it's assuming the same parameters in every node. But then we decided to also also use this other approach of heterogeneous model, which has been introduced more recently, that allows these local weights to be variable across different nodes. And we did this based on a combination of 6 fixed maps from neuromaps. And here, because you have more freedom, we have more free parameters, and it's more difficult to fit. Okay, then we have our model, we have a set of candidate parameters, then we plug it into the model, solve the model, or do a numerical integration, and then get a simulated bold, right? And then on the other hand, we have the empirical bold from the same subject. And now the question is, how well did we do, right, the simulation? And we want to compare these two, simulated and empirical bold. And we do this through features derived from these. One feature is functional connectivity. And we want the functional connectivity of the simulated data to be maximally similar to the functional connectivity of the empirical data in both the patterns, therefore maximizing correlation, and also the magnitude, therefore minimizing the mean absolute difference. Then functional connectivity is a time average measure. It doesn't consider dynamics. We wanted to also consider dynamics. Therefore, we did this. We calculated dynamic functional connectivity of the signal by using a moving window and then calculating functional connectivity in every window, which will lead to this GIF, which is dynamical functional connectivity. And then we can correlate the patterns observed at different time windows into a matrix which is called functional connectivity dynamics matrix. Now, the distribution of values within this matrix can describe the state of how much recurrence there is, how much stability there is in the dynamics. And we care about the distribution, right? So we do the same thing in the simulated data, and then we look at their distribution and see how close they are. So therefore, we want to minimize the distance of the distributions of functional connectivity dynamics matrices. So we have 3 goodness of fit features, and we combine them into a single measure of goodness of fit. And then so this was for one set of candidate parameters, right? We want to repeat this step over and over for different sets of free parameters until we get the best goodness of fit for this particular subjects. And AKA, we want to do model optimization. And we did this using covariance matrix adaptation evolution strategy, or CMAES, which is an evolutionary paradigm. And you can see here that on the scatter plot, each of these dots are random points through the parameter space, and each of them is a simulation, and their color shows the goodness of fit. Now see how the algorithm finds its way towards the point of maximum goodness of fit, the yellow point, right? Okay. Okay, so a quick note on implementation. Here we had to do a lot of simulations because we had many parameters, we had many subjects. Each of them had to, it was crazy, like the number of simulations that we had to do. And we wanted to have a very, very efficient implementation for the simulations. And we came up with a GPU implementation of the simulations. And by the way, we did all of this on the Jülich Supercomputing Center. And the GPU implementation allows us to parallelize across nodes and simulations, something that we cannot do on CPUs. And that enables crazily fast simulations that is much, much more faster than CPUs. And that enabled this hyper-parameterized model with 15 free parameters, which is normally otherwise not possible on a CPU, or very difficult. And it paid off because we can see that the goodness of fit in the heterogeneous model is considerably better than the homogeneous model, right? And therefore, I will continue also to talk about the heterogeneous model. Okay, I have to skip a lot of results. Therefore, we had this, so we finally have our subject have the optimal parameters, right? We plug it in, and this is the individual's model of our current subject, right? Then we look inside the model, look at the firing rate, excitatory firing rate, as a measure of EI ratio, because it's also intuitive. If you have more excitation, firing rate is going to increase. If you have more inhibition, it's going to decrease. And then average it over time. Therefore, we have one of these maps per each of our 627 subjects. And then let's look at this age variation, and we can see this interesting pattern. We can see decreases in association areas and increases in somatomotor and visual areas. And if you look at the significant ones, like these are the significant ones after FDR and after everything. And then one other way to also look at this distribution, you can see age effects by the different Yeo networks. You can see again positive effects in somatomotor and visual, and negative effects in association networks such as limbic frontoparietal network and default mode network. And I wanted to show this, that if we average firing rates across all the nodes and look at this range variation, we don't see anything. So we will miss out all of this. And I have a bunch of sanity checks which I don't have time to get into, but longer story short, I wanted to make sure that these results are not affected by changes of a social connectome through age, which they are not, okay? Or not, they're not too, like, it's not entirely that. And then I also wanted to make sure that these are not just random data that we have, and repeated it in another run, not another run of fMRI, but another run of optimization, and then observed a great deal of stability of the findings. And like, I think I should escape on my conclusion. So we showed that this decrease in association areas is it very well matches a lot of findings in the literature, and we're excited about that. And so we want to also do a bunch of more things because it's also the very simplified models, and we want to do more stuff. Never mind. And yeah, thank you very much for your attention and to the co-authors, and yeah, I'm happy for the questions.
Well done at the end. Do you have one quick question from the audience? If you don't, I have one quick question for you. So this was showing development primarily.
Yes.
Does the opposite happen with aging?
Would be curious about that. I don't know. Yeah, could be one other next step that I have.
You got something to do for the next year or so.
Yeah,
Great.
Thank you.
Thank you very much.